All right. Tonight it is my pleasure to introduce our speaker, Dr. Lila Fishman, a professor at the University of Montana. She is going to give you her full introduction. So with that, I will just say welcome, Lila, and everyone. I hope you enjoy tonight's presentation.
Okay, so I'm going to switch over to share my slides, but since this is my first time talking to you all, despite having lived in Montana for 2 decades now, and being a plant person the whole time, I thought I would go ahead and introduce myself and some of my history in studying plants off the bat before we get into monkey flowers. So,
And
can you do, I think
sorry, I'm just, I'm getting out of
there's a lot
my,
of mouse action, but I need to share, I'll share my slides.
let's see.
Share. Um,
Sorry, Leela,
no
let
worries,
me just
no
get
worries.
out of here. I'm stuck in my, okay, there we
As long
go.
as we've got monkey flowers on the screen, it's
Stop
fine.
share. Okay, there we go. I'm
Okay,
sorry.
so no worries. This is smoother than many classes. So
Okay, well, that's
let me get back so I can see you guys a little bit up here. Oh no, not that. Sorry. Let me go back to the speaker view, and I'm still seeing your slides and my slides. Can you guys see my slides now, please?
we can see your slides. They look
Excellent.
great.
And they look
Yeah,
good. Okay, great. And
yeah,
if
they look great.
there's people keep popping up. So I've been admitting them while you were talking, but if you could admit
Oh,
them while I'm talking,
thank
that'd be
you.
great.
Yep, I'm on it.
So, um, so, um, yeah, so gosh, it's nice if I can see at least one person's face. So I'm talking to somebody. I'll see my face at least. That helps somewhat. Oh, there we are. Okay, good. It's just, otherwise I feel like I'm speaking to the void. Yeah, so my goal for today, again, is to tell you a little bit about what I know, which is mostly about evolution and in monkey flowers. And I wanted to start by just sort of talking about my path into being an evolutionary genomicist. It's not where I started, since genomics wasn't really a field when I started out as a scientist. Then I was going to spend a little bit of time talking about monkey flowers in general and the former genus, maybe still genus Mimulus, as a model for diversity. It's a model system for a lot of research groups, actually. But it is a taxonomically complex group. So I was just going to sort of survey over that. And then I was going to talk a little bit about two more active research vignettes that we're doing now. And again, hopefully there'll be lots of times for questions. We studied lots of different things and I'm interested in lots of different questions. So these are just two of the many things that we study. So to tell you a little bit about myself, I always feel a little guilty when students ask me, like, how did you get into plants? And I'm like, I've always been into plants. So I grew up in really, really off-grid Northern California. So can you guys see the mouse if I'm moving it on the screen? Anyone? Okay,
Yes,
well,
yes,
Yes.
we
the
can.
star over here on the map, that's the Lost Coast of California. The Lost Coast just means an area where like the freeway doesn't go along the ocean. My parents were hippies. They went back to the land. They moved us out there when I was 4 years old. And this is the lower picture here with this is our Rosa omiensis or Sericea terracantha omiensis. It's a Chinese rose. But we moved out to the middle of nowhere, and my parents had a nursery growing roses and antique apple varieties. So I very early on was spending a lot of time pruning and working with plants. And it's also just an amazing place in terms of the diversity, like much of California, but particularly this is the land of redwoods. The upper, for me, right corner is the Council Madrone, which is huge, probably the biggest in the world, actually. It's broken now, unfortunately. Madrone tree, so really high beta diversity, so lots of different kinds of habitats, lots of different kinds of plants, kind of region, as well as all the horticultural plants that I was sort of exposed to on a daily basis. So I got interested in plants out there in that really diverse landscape, and then went away to college back east at Swarthmore College, which I went there because actually the whole campus is an arboretum. It's the Scott Arboretum of Swarthmore College, and this is so a very different kind of habitat. It's my first time on a plane. Went back east, And I worked the whole time I was a student there in this cute little building down on in the corner, which is actually an old astronomy building. Making labels for the horticultural collections on that campus. So and you know, I thought, as I say, always been interested in plants, but I thought I was going to be an ecologist. So then I went to graduate school um, at Princeton to try to understand how so many different kinds of tropical trees can coexist together in a place like Barro Colorado Island in Panama. So I spent several years doing field work down in Panama on these, they're called the 50-hectare plots. There's a very intensive research effort down there, spearheaded initially by Steve Hubble at Princeton when he was there, where every single stem on this 50-hectare plot is identified and labeled, and that's a lot in that habitat. So very, very dense forest. Trying to understand patterns of seedling recruitment and how they might be affecting the maintenance of diversity through increasing spacing between adults and younger plants. So that was super fun and interesting, and a lot of interesting diversity down there. But I quickly came to realize that community ecology might not be for me. I also got terrible chiggers. And so what I realized, and as it sort of moved me towards becoming an evolutionary biologist, was the sort of flip side of diversity, which is that if you have, 300 species of woody shrubs in a 50-hectare plot, any given species, an individual might be very far from another member of that same species. These are aerial views of Barro Colorado Island. It was a mountaintop formed when the Panama Canal was they flooded and formed Lake Gatun, and so that's why it looks like a mountaintop. And so when you look at these distributions, some individuals may be very, very far from a member of the same species that could be a mate. And I got really interested in that, first from an ecological perspective, and then from an evolutionary perspective. Particularly, I started working on these psychotrias shown here, which are shrubs that are related to coffee and cocaine plant, coca plant, actually. That also turned out to be very difficult. Fortunately, I had a good fellowship and was able to switch projects again. So good lesson for young people. You don't have to know exactly what you're going to do before you do it. And I switched to working on the sort of evolutionary consequences of sorry, trying to move this so I can see a bit more. The evolutionary consequences of sort of ecological mate limitation. So if, again, density might cause that, competitors for pollinators might cause that, low pollinator service might cause that. And so I started to be interested in how differences in plant mating systems, and I guess I don't know how much people, I mean, you're all planty, presumably, but I don't know how much you know about mating systems. So forgive me if I tell you things you already know. But plant mating systems just describe the variety of ways in which sort of sexual reproduction, so the production of seeds, in this case through pollen and ovules, joining happens. And one of the really common patterns in flowering plants is over and over again, you see showy, bee pollinated, hummingbird pollinated flowers have close relatives that are highly self-pollinating. And this happens very, very commonly in flowering plants. And I got interested in the processes that generate that, and whether self-pollination was generally evolving, or specifically in some cases evolving, to ensure against reduced pollinator service, whatever the cause. And so I then switched to work on this sort of very general question of why plants evolve self-pollination. It's still a very active question of research. So this is back in the early 90s. Working on these little tiny Caryophyllaceae, then known as Arenaria. I think they're probably Minuardias now, that grow on granite outcrops. Oops, mouse went the wrong way there. In the southern, southeastern US, so like in Georgia and Alabama and the Carolinas. And this is a really fascinating system. So Robert Wyatt, who was down at the University of Georgia, had already been working on it for quite some time, and had noticed this really intriguing pattern where you have Arenaria uniflora on these granite outcrops. which are like Stone Mountain in Georgia is a world's biggest granite outcrop. It's just a big chunk of rock, sort of a mountain remnant that sticks out from red clay and scrubby pine landscape. So they have these very unique plant communities on them that are sort of remnants of higher elevation communities. In any case, most of them were the relatively large flowered, but not super showy outcrossing arenaria uniflora. But in areas on the edges of the range here where it's in this hashing on our very antique figure, we see that the flowers get even smaller, and they become temporally and spatially the sexual parts touched to allow the plant to self-pollinate. And that is occurring in areas of range overlap with a larger flowered Arenaria glabra. I say none of these things are super large flowered and showy the way Mimulus are. One of the reasons I switched. But this is a really intriguing pattern, and Robert had hypothesized before I started working the system that basically competition for pollinators between the outcrossing uniflora, in the central part of the range, and when it moved over to the range edges where Arenaria glabra was occurring, the insects were preferring that, and that's why it involved selfing, because it was basically never getting pollinated. But if you self-pollinate, you don't need to wait around for a pollinator who's off visiting some other plant. So hopefully that makes sense. So I did this is just title of one paper. We did a lot of different work on it. But we actually found that is actually the case. So selfing, those ecological interactions between those two species, so the glabra and the uniflora, appeared to have influenced one of them to basically back down from competition for pollinators and just become a selfer. So sort of a complex system here, not just loss of pollinators, but presence of the other species. That's what I'm showing in the graph. So outcrossers lose in the presence of the other species, so lower fruit set, and the selfers are not bothered. And I did this all with like arrays on outcrops in the field. But there was actually an even further twist, which is that it turns out they actually can mate with each other to some extent. So pollen from the big flowered species, the Arenaria glabra, can get on the outcrosser uniflora. And when it does, the pollen tubes grow all the way down. And you might even get a few seeds, but you don't get any viable seeds. And so it's not just competition for pollinators. There's this deleterious pollen gene flow or bad pollen gene flow from the one species to the other species appears to have, I'm always caveating because I'm a scientist, but all the evidence suggests that the self-pollination has evolved not just as a way of assuring its own reproduction, but of avoiding deleterious pollen flow from the other species. So that was super cool. We also looked at lots of other things like inbreeding depression and the pollen export and how that was being influenced by the shifts in flower size. And this got me really, really interested in trying to understand these kinds of evolutionary processes. This work is all sort of not genetic in the sense that, sorry if there's fruit flies in my office. His work is not genetic, but it's very much about thinking about the processes that move species from one endpoint to another. And that is what got me thinking about how we can reconstruct the path to those different endpoints all along the way, rather than comparing only the extant species. So if we compare the selfer to the outcrosser, they're very different plants at this point. They're different populations of the same species, or some people call the selfer I think it's alabamensis, so Minuardia alabamensis now. And so those are endpoints, but it took a lot of genetic steps along the way. And really what we want to think about if we're going to understand the evolutionary process is, at least for me, is how each of those steps influences fitness. And so that's how I started moving into a more genetic approach. I just, these are new slides for me. So sort of general take home from that, though, is just that it's really, and this will come back a little bit when I talk about Yellowstone work as well, is that we can see that mating systems are very, very important, and that selfing can provide reproductive assurance. And these are just some examples of recent work in the same field using sort of similar approaches that also show that plants use selfing as a way of assuring seed production in the face of both past kinds of challenges, like being in contact with another species that hogs all the pollinators or puts pollen on you that's not compatible, but also in response to more recent anthropogenic change. And so the images I'm showing you here are just from a very recent review paper emphasizing that we need to think about plasticity in plant mating systems in response to climate change, as well as evolutionary responses, and that's very true. And then the picture of the pansy, which hopefully you guys can see, is from a New York Times article about a year ago. Olivier Cheptou, French researcher, showed that using sort of resurrection approaches, that pansies are evolving to become more selfing over the past couple of decades as pollinators are becoming more rare. So it's not, for an evolutionary biologist, it's not a surprise that evolution happens, because it always is happening. But this is a case where they've been able to really nicely demonstrate that in some number of decades, you can have a measurable shift in that mating system. Hopefully now that you guys can see that title, and those of you who are savvy will realize that there may be also it's an article from Carl Zimmer, who's normally super good at doing science writing, but I feel like I needed to clarify this, and some of you may have seen this. So the title here is "Flowers Are Evolving to Have Less Sex." And I just want to emphasize, since we're going to be talking about mating systems throughout, that self-fertilization is not sex. It is, is sex. So it's not clonality, which would be making an exact copy of yourself. It's bringing male and female parts together within a single flower to produce a new combination. And this is why inbreeding depression is a problem. This is why we have inbreeding taboos in humans. Many animals have behavioral avoidance of inbreeding. Many plants have genetic avoidance of inbreeding or morphological avoidance of inbreeding, because you're not just making a copy of something that's perfectly good, you're remixing it, but you're not getting any new variation, and that can reveal deleterious variation. So selfing is sex. It's just sex with the same flower. And again, you guys probably know this, but in case you don't, it's really important to remember whenever we're talking about plants, they're very different, especially flowering plants rather than conifers. They're really different in that about 90% of them have male and female parts right in every single flower. So they have the option of selfing. And if they're not selfing, it's because they have adaptations to attract pollinators, to keep the male and female parts apart, or to do other genetic incompatibilities that prevent selfing. So really important to realize that, and we'll come back to talk more about these kinds of mating system shifts later on. Okay, so I'm still in the introduction. That was where I was as a grad student, really excited about mating systems, thinking I should become more genetics. I was very lucky as a postdoc to go and work with the man illustrated here, John Willis. He's still a professor at Duke. I started working with him at the University of Oregon, and then we all moved to Duke. And really, at that point, for me, it became very clear that in order to understand, again, the sort of many steps along the way to complex diversification, we need to understand not just the endpoints, but the steps there. So this is basically taking the same kind of approach as Mendel would have. So crossing plants that are different, as we saw over here. So we can cross a tall and a short, get all tall. This is what happened with me and my husband. He is tall, I am short. Our kids are like both 6 feet tall. I'm like 5'2". So no segregation yet, but we have dominance here, so we get the all tall, but then we can segregate out the variation in the parents in the offspring. And so we do that. That's a lot of what we do in my lab is generate those kinds of hybrids, that same way that Mendel would have, but then add on a layer of genomics where we're then identifying the particular loci using methods that we're not going to go into here, and you don't need to know right now, but you can ask about if you want. Then we can understand not just the total shift in phenotype. So here I'm showing Mimulus nasutus, which often doesn't even open a flower. It's very closely related to and crossable with Mimulus guttatus, which makes a nice big bee-pollinated outcrossing flower. And the graph below there is showing at least 24 regions of the genome that contribute to the differences in flower size between these two. So each of those does a little bit of the difference, and each of them might affect one trait more than another trait, or the position of the anthers as well as the corolla length, but they don't all have to do things exactly the same way. And it's really those individual steps that natural selection has been acting along throughout the diversification process. So I should say John Willis was really early on in sort of seeing natural variation as a sort of tremendous pool of information about diversity that's both useful from sort of a crop plant perspective, but also really fascinating from an evolutionary perspective. And so started really applying the kinds of approaches that people had been using, so prior to about 20 years ago in it just in a very few crop systems, or the model plant Arabidopsis, which is a little mustard that people use for genetics, applying that to Mimulus. And it so happens that we and others have been applying that approach to Mimulus for a good long time. I feel like I should stop and let you guys ask questions, but let us keep going until I get to the end of this section at least. Slides are going too fast. Okay, so that gets us to the University of Montana. Maybe we'll stop after that. We can chat if need be. So I came here about now 2 decades ago, and our research is really devoted to understanding plant evolution from a genetic and genomic perspective. So again, that's sort of picking things apart to understand how they got to be the way they are perspective. We mostly do that in Monkeyflowers, as I'll talk about, but we're also sort of engaged in lots of other kinds of planty things on campus, because there's not a ton of, because we like plants, but also because there's a finite number of plant folks here. We're not the ag thing. So I just wanted to highlight, as part of my sort of introduction to me, 2 other things at the UM campus that I'm very involved with. The first of these is the herbarium, which has been sort of an on and off and on again. It's currently on. So the herbarium here is really nicely curated now by Giovanna Bishop. Hopefully you guys will get to hear from her at some point as well. She more works on things like lichens and mosses, small things, not the big flowery things. But she's been really, the past couple of years, just revitalizing the way that the herbarium is run, and we have a much greater presence there. So that's on the dry plant side, and something that's very useful to everyone statewide. I should say, years ago, we databased all those collections, part of a grant that I wrote to NSF, and they've been added to the Consortium of Pacific Northwest Herbaria, and we're currently working towards completing that with a new grant, with the new, led by the new head of the Berkey Museum, Carrie Tribble. So that's super exciting, and hopefully you guys will all get to meet Giovanna. The other person who I think she might be on here. We also, and this is more for people right in Missoula, but if you are interested in plants and might need to grow them for some reason, we developed now about 5-10 years ago a plant growth facility that includes both greenhouses and controlled environment growth chambers. And that is very wonderfully run by October Moynihan, Dr. October Moynihan, I should say, who manages that. And so both of these are things where, if you are in this part of the Montana and interested in plants, they're really nice resources and things to know about. Okay, so that is my long introduction to me, things that I care about. Were there questions in the chat yet? I don't want to, can't tell.
I don't think
Okay.
quite
No,
yet.
those are people checking in with their locations. Okay. Did anyone have anything they burningly wanted to ask before I proceed onward? No? I can't see you guys, so
It looks like
too many things on my screen.
not yet.
Okay, great. That means we shall go forward into Monkeyflowers as promised. Oh, sorry. Before we go to Monkeyflowers, sorry. I give either research docs or teaching, and this is kind of neither. So I'm curious to see how it goes. So again, what we're interested in as plant evolutionary geneticists, or evolutionary geneticists in general, is sort of the nature of variation. And that really goes from the individual level up to the beyond species level. And so in my lab, we tend to ask about what maintains variation within population, both visible variation, so phenotypic variation. We have, for example, in Oregon, we work on trichome polymorphism, where some plants are covered in glandular hairs and other ones don't have any at all. And that's under the control of genes. It may also respond to the environment in various ways. And like, why? Why are there some that do one thing and some that do another in the population? One answer is obviously drift, but it's often some form of natural selection. So we focus a lot in trying to understand variation within populations at the individual level. Then we sort of also scale up to think about variation among populations. So local adaptation, divergence in that is often in response to environmental factors. So a lot of our work is focused on that sort of middle question of adaptation. And then lots of work focused that kind of comes out of the adaptation in some cases, but not in others, focused on understanding the evolution of species, so the buildup of reproductive isolation that lets us define species. And that includes both the visible things like differences in pollination syndromes that, cause plants to never even mate with each other, as well as things like hybrid incompatibilities, like you would have in a mule, where you can hybridize two plants, but then the hybrids are messed up in some way. And so obviously, I'm not going to tell you about all of this today. I'd love to, but I don't have time. So I'm really going to be focusing on this middle section after we talk a little bit more about monkey flower diversity overall. Okay, so this brings us to monkey flowers as billed. And this is actually, this illustration is from a review article in the Science, which is the sort of top magazine for all science, all fields. Again, as I mentioned, there's actually quite a lot of us that study monkey flowers from an evolutionary perspective. For one reason is that they're super, super beautiful and diverse. The other is they're very tractable. And so again, this is not just me. This is a sort of a general group of researchers who are interested in using monkey flowers as models for understanding plant variation. So I'm going to tell you a couple little stories from that today. But I think because we are sort of using the common name here of monkey flowers, but as scientists, we often need to be focused on the scientific entities. I'm going to spend a little bit of time, not too long, talking about what even are monkey flowers. And so, because again, we wouldn't ordinarily use the name. So I'm just going to walk through some, mostly with pictures of my very own or my collaborators, where I've gotten permission a few times with photos that are from places I haven't been able to get to, unfortunately. I'll try to acknowledge those as needed. About sort of some of the diversity of monkey flowers. Again, we're getting genomes for many of these taxa, and so it's really a work in progress to understand how they're related to each other. But I'm going to give you kind of a cartoon of how the people who study evolution in Mimulus are thinking about evolution at different scales in the system. So, okay, so to start, at the very beginning, and again, I should say I'm not a taxonomist. I'm, as someone who studies speciation, I'm very comfortable with well, to mention species complexes. So these are groups of organisms that might be morphologically divergent, but can still hybridize, but might be partially isolated. I'm very comfortable with speciation being a process that takes a long time. It's rarely instantaneous, but we need taxonomy to communicate about the plants. And so the phylogenetic tree I'm showing you here is from Dina Grossenbacher, who's at Cal Poly San Luis Obispo. She's an amazing natural historian, took all the images of monkey flowers and figures, she knows a ton about them, about their field biology. We've done some nice collaborative work together on Mimulus bicolor, or erythrinthe bicolor, out in California. So I'm sort of going to go through the groups here. The names that are on the outside here are the sections of the former genus Mimulus. The phylogeny is based on a few genes. It's not probably that wrong, but it's not the complete picture of the variation. And so I'm just going to kind of walk through the diversity and then ending with a focus on the ones that we primarily have here, which are also the ones that I primarily work on. So way back, these guys were members of, they're in the Lamiales. I feel like I had a picture of the Lamiales. Sorry about the slides. Yes. Yeah, so way back they were in the Scrophulariaceae. And this is just a cartoon from the internet of the former Scrophulariaceae, which are, things with nice bilateral symmetric tubular flowers. So mints and these nice bladderworts, the Latubulariaceae, you got a foxglove, all the Orobanchaceae like Castellea and other hemiparasites, Bignoniaceae and Acanthaceae, all of these things. Many of the members of these were sort of mixed up, some of them globbed into the Scrophulariaceae. Paul Beardsley and Dick Olmsted, quite a while back now, basically figured this out and moved Mimulus into Primaceae and many of these other groups that had been in Scrophulariaceae into other families. And so that then made a new family, Primaceae, which consisted of Mimulus, Prima, which is a monotypic genus, and there's 2 subspecies of 1 species, and then a couple of South American taxa, Hemikina and Leucocarpus, that were sort of interdigitated within the Mimulus. So clearly not perfect yet. And that then is sort of where this is. So we have Mimulus in the Primaceae, and now we're thinking about variation within that group. I'm not going to talk about the Central American ones, so we're sequencing them. So then the flora of North America, oops, sorry, I don't know why the slides keep moving without me. There we are. So then with the flora of North America, and I know Pete hasn't yet updated the flora of Montana, probably at my request on this. Guy Neeson, who was charged, who hadn't worked on Mimulus before, but was charged with redoing them, and is a really good seer. He sees variation very well. He and then others since did sort of 2 things. The first of these is to split Mimulus into 3 genera. So including those, not including those 2 South American ones. So now we have Mimulus, Diplacus, and Erythranthe. That's just a little hard to say. And then he and then Naomi Fraga, who I'll show you a picture of a little bit, who works a lot with the more Californian ones, have then been also naming many species within particularly Diplacus and Erythranthe. Okay, so and a lot of that is there's a ton of cryptic variation. There's kind of not so cryptic variation. There's there's a lot of activity in this group. And I say I'm going to use this sort of phylogenetic structure to walk through. These are how they're related to each other. I'm going to mention chromosome numbers because they're kind of important in describing groups. But we're sort of going to walk through each of the key groups here. Starting, I should say the sort of complicated thing is that there's a very large community of monkeyflower researchers who've been working on Mimulus since long before these name changes. Often in these kind of ambiguous groups where we have things that are maybe species, maybe not, where we're interested in understanding the processes that generate the variation as opposed to putting things into discrete types. And so there is sort of a fair amount of back and forth in the community about use of the new names, and many of us, including me, still use Mimulus names when we're talking about, when we're writing scientific papers, simply because, not because of genera are arbitrary, so it doesn't really matter what the genera are, and theirs are mostly, they make good sense. It's more that the species level, there's been a lot of naming of species that doesn't necessarily accord or isn't yet supported by the depth of phylogenetic data that, or doesn't accord with our species description. So I'm happy to talk more about that later, but we're still sort of in a state of flux. Eventually there'll be enough data that we'll know what is a species, and then it will be unambiguous. But so I'm going to continue to refer to things as Mimulus throughout, but I know that many other people don't. But I don't care to argue about it right now. The other thing, so as you say, we're getting very large numbers of genomes, so not just the kinds of Illumina sequence that you would get from individual sequencing, but whole contiguous, what we call telomere to telomere or full-length chromosome genomes for about 50 Mimulus. That is being done by the Department of Energy, at least as of last week. Who knows what they're doing this week? So the Joint Genome Institute of the Department of Energy, they early on sequenced a Mimulus guttatus for us that has been very, very valuable to this entire community of researchers. But now they're doing all the highlighted taxa across this group, assuming we can get enough tissue from them, will have complete genomes, and that will provide a really, not just a picture, that will provide a picture of both the sort of sequence diversity and evolution, but also of sort of the chromosome scale. And that will be a fairly unprecedented genomic resource for plant biology in general. And the reason the Department of Energy is doing it is because Mimulus are tremendously adaptive and live in many kinds of difficult places, and they're very interested in understanding the basic biology of plants from a biofuel perspective. Okay. So And just again to emphasize this, this is one of the reasons we keep using the name Mimulus. This is Mimulab. This is a Slack workspace. I think there's over 200 people now. This is an old screenshot of basically people all around the world who work on monkeyflower evolution. We have like a every other year meeting, workshops and things like that. So they're not just beautiful plants you see around you. They are really a model system for understanding plant variation from lots of different across lots of different axes of different kinds of variation. Okay, so yeah, so now want to just show you some pictures of different monkey flowers. We have pretty good diversity here, but I just want to talk about some of the diversity of things in the sort of Mimulus old sense. And this composite is from Jay Sobel, who's at SUNY Binghamton. He's a collaborator on a number of projects with us. And so this is showing sort of some of the overall diversity, but I'm just going to walk through each of the former sections. And again, this diversity is fascinating, but it's the diversity combined with the close relatedness of things with very, very different biology that make them super valuable, not just as a picture of diversity, but as a way of understanding the process of diversification. So actually the A picture here, which I think is not Jay's, though all the other ones are, it's actually a picture of copper mine tailings in California. So like we have here, and that's been going on for about 100 years in California, and offshoots of Mimulus guttatus. So both populations of Mimulus guttatus, but also a sulfur called Mimulus cuprophilus live, and pardalis, live on these copper mine tailings where almost nothing else can. And that has been a really sort of, but then they totally can hybridize with, are not visibly different from in a greenhouse from plants that grow right nearby that are completely intolerant of copper. And so this provides a way of asking, well, how do they do that? How have they adapted to deal with the copper thing from a physiological, from a developmental, from a genetic perspective? And so it's the diversity with sort of not sharp barriers between things that is actually the most valuable from a scientific perspective. The diversity is beautiful from just a straight up perspective. Okay, so I'm going to walk through some of that diversity. So starting basically on the East Coast and at the most distant from the ones we have here in Montana, and the ones I work on. So the first group is the actual things, only ones that are really still called Mimulus under the new genera. So this is the Ringens group. There's obviously a bias because European colonists are naming things and they are coming from the East. So the types for the genus is in the East. This is a plant of the wetlands. You can see down on the lower right, can be quite a large bog plant. There's Ringens, there's also Elatus. They get a little bit into Eastern Montana here. They're generally blue flowered, but they're also, weirdly enough, seem to be allied with a number of taxa in Australia and one South American species, Mimulus strictus, which is the inset, this white one. And again, we haven't sequenced all of these yet. That's in progress. But morphologically, it does look like there are basically, Eastern North America, Australian, and the South African species that are quite similar. And there's, I think, 4, 5, 6 genera that appear to be sort of close to this Mimulus ringens group in Australia, including these color polymorphic Uvidalia. So, and they're Michael Barker, I think, studies those primarily. So these are very far away from the ones that I studied. So it's hard to even line the chromosomes up. But these are the remaining true Mimulus. The next group, which we don't have a ton of here, but you might encounter in Western North America, is the Diplicus group, starting with what we call the Mimulus aurantiacus, or oops, sorry. Every time I move my mouse, bad things happen. This is the Mimulus aurantiacus. Sorry, I need to say it more slowly. Group. These guys are shrubs, so they're woody, except for Mimulus clevelandii, which is the yellow one. I'm going to be very careful with the mouse here. The yellow one is herbaceous, so So a number of researchers are using that as a way to look at the evolution of woodiness. Um, so, and like the ones that I'll tell you about that we work on, these ones also show tremendous floral diversification. So on the map of California, all those different flowers, actually, many of these used to be called Mimulus aurantiacus or subspecies of, but you can see there's quite a lot of floral diversity with Puniceus, and several of the others being primarily hummingbird pollinated, and then the paler ones being primarily moth pollinated. And obviously that's important from a speciation perspective, but also from a sort of fitness perspective in terms of attracting and efficient pollinators. Um, and the sort of the pretty flowers down in the lower right there. This is just these are hybrids that are very easy to generate, even occur in nature, primarily in the area around San Diego. That where you can basically all of these things share alleles, so they sort out into these species on the landscape. But if you would go to any individual site, you might see mixtures of flowers like in the picture. And this system has really been developed, and sort of the story continues to be told by Matt Streisfeld, who's a faculty member, professor at the University of Oregon. So these are super cool. And again, this is not all, and these are the ones that are sort of the type for, they used to be called Diplicus, and so now they've been renamed Diplicus. The other group within Diplicus, and I'm not showing you all, but just some of the diversity. The other group are what were sort of formerly sections Eni and Unanus. And again, I'm not sure I'm saying this right. So this is things like Mimulus nanus, which is this middle bottom one. You can see really pretty. The stigmatic surface is the same color as the bright pink there. This one is tremendously floral diverse. They're mostly in relatively dry habitats, which is different from the Mimulus. We'll hear about it for in a moment. And they often have these even color polymorphisms within populations. So the two color picture, those are actually members of the same species. And it's a single gene that controls that pink yellow polymorphism. They also have these two totally cool sort of moth pollination syndrome species with these almost radially symmetric flowers and this very, very striking dark center. We just finished sequencing Mimulus pictus, which is the inset one with the really nice patterning on it. And so there's a lot of interest in trying to figure out how and why this has occurred. The yellow one down on the bottom is another cool one. It's Mimulus vandenbergensis, which is restricted to basically sandy dune areas in Vandenberg Air Force Base in Southern California. So these are sort of desert ephemerals, often annuals that are just sort of coming in when there's heavy blooms, when there's rainfall. And this group has a lot of undescribed variation in it still, I would say. There's been a lot of new descriptions going on, but there's still a lot of undescribed variation. And Naomi Fraga, who's down at the California Botanic Garden, used to be Rancho Santa Ana. She's really the expert on this group. And I don't even know all the new names. And she's also doing really amazing conservation work with this group. The link, I don't know if you guys will be able to get it later, but there's a really nice New York Times article about how she's doing conservation in the Diplicus, as she would call them, down there. But anyways, a really good person to follow if you're interested in this kind of wildflower diversity. I put her Instagram handle there as well. Okay, so these are the Mimulus that are kind of far away from the majority of ones we have here. We have a few of these Mimulus nanus-like ones. So you go down to like craters of the moon, but they're the mass diversity we have here is in this next set of plants, which would now be in genus Erythranthe, or Erythranthe, as I like to say, but I've been told it's Erythranthe. And these are the sort of wet-loving Mimulus, like ringens. They tend to occur wherever there's seasonal standing water at the very least. So let's look at those. Yeah, so these are ones that you will have seen if you're in Montana, if you've ever been to Glacier. So the first group of these is the Mimulus cardinalis or Mimulus lewisii complex. And this was what was section Erythranthe. And these guys are again are a complex in that there's if you look at the phylogenetic tree, when I try to use my mouse, it makes the slides move around. If you look at the phylogenetic tree, the pink are Lewisii up in the upper corner there. And then we have Mimulus cardinalis, that's all the red. The Arizona ones are in orange there. And then there's these two other species here, Verbenaceous and rupestris, that are also hummingbird pollinated. And then Mimulus parishii, which is a tiny flowered little pink selfing one from Southern California. And for a very long time, these guys have been a model system, not just for this really sort of dramatic floral divergence, which is again between species that we can still hybridize them, and they still hybridize in nature where they're in contact. So these look very different, but there's not that much that separates them. So obviously we have maybe not obviously we have the bee pollinated in the big pink ones. We have the two sets that are hummingbird pollinated, and then the selfer. And these guys have been since the 1950s, actually, long before people started doing genetics, been a really important model for speciation by pollinator shift and also by elevation. So in the Sierras, the red Mimulus cardinalis, so we don't have that here, but it's common throughout California and Arizona. It is hummingbird pollinated, occurs at lower elevations. Mimulus lewisii, which out there is a paler pink, and it might be known as iridescence. Then the lewisii we have here, say, Glacier at high elevation, but it's similarly high elevation, bee pollinated, and they sort of have this narrow band of range overlap in intermediate elevations, but then are sort of in contact all along the Sierras. And some of you may have heard of the classic work of Clausen, Keck, and Heisey. They're sort of the folks who developed the ecotypic idea of variation among plant populations, lots of reciprocal transplants, moving plants between high elevation and low elevation sites in California. And among many other things, they also worked on yarrow and potentilla and a bunch of things. Heisey, who was part of that group, then worked quite a lot on these Mimulus, doing transplants and trying to understand what were the barriers that kept them apart in areas where they were in contact, and what were the mechanisms of a local adaptation that were leading to speciation. Okay, so with the pollinator shift and the elevational restriction being the key things. And that then sort of was taken up by Doug Shemsky and Toby Bradshaw years ago now, in like the 80s, and did some very, very influential work hybridizing Mimulus cardinalis with Mimulus lewisii. So this is a very classic Nature paper from 1995, where we have the hummingbird pollinated one, the bee, that's C, the hummingbird pollinated one, that's A. I'm sorry, the bee pollinated one, that's A. You cross them, you get something that's intermediate, but more lewisii-like. And then if you hybridize them, you sort out variants that look like J and L. Some of them look almost just like the parents in all dimensions, which is actually not really what we'd expect. That's certainly not what we found for the work we did in other Mimulus. But things like nectar volume, and which is very important for hummingbird attraction and hummingbird rewards, they keep coming back, as well as the color, as well as the flower shape, would all stay very tightly packaged together, even in these hybrids. And so this sort of started Mimulus as a model system in a more general way. And it continues to be. So this is just a review, sort of a pastiche of work by my collaborator Yao Yuan, who's at University of Connecticut, and he worked with Toby Bradshaw back in the day, but now has really turned this into not just a model system for sort of cool ecological and evolutionary variation, but straight up developmental variation, which again I'm not a developmental biologist or a molecular biologist, but he is. And so they recently, this is the cover of Science from about 2 years ago, figured out the actual very specific genetic change that produces the carotenoids that make you go from being pink to being red. It turns out to be very tightly linked to genes that change your anthocyanins. So there's what's called a super gene of multiple genes, so producing multiple different proteins, but packaged together on a chromosome. And again, just we don't need to dig too much into the genetics, but a really interesting picture of how not just individual mutations in individual genes, but the whole genome architecture can contribute to the evolution of complex traits like pollination syndromes, where you don't want to be red but not make nectar, and you don't want to be white because then you have that doesn't necessarily fit with any of the pollinators. You need to sort of leap between the set of traits that is Mimulus lewisii and good for bees to this whole other set of traits that is, makes you good at being hummingbird pollinated. And that might involve genes all over the genome that you then need to sort of coordinate together. And that's hard to do in Zimpatry. And so Yahoo's really been focused on basically figuring out every gene that influences flora morphology in Mimulus, knocking them out one by one, but also then using the natural variation as a guide. So to say this is really nice from my perspective. I'm a little bit more on the field variation to greenhouse, and he's sort of more only in the lab, but it makes a really nice set of tools for us to try to understand the variation. Okay, so that's Erythranthe group. And again, these guys have 8 chromosomes. So I should say we also work on this group in collaboration with Yahoo, but also with other folks. And I'm not going to dig into the details here because again, I don't have time, and it may require more genomics than you probably want to have. But it turns out that the system is really, really interesting, not just for that simple pollinator shift, but that as we've started looking at the sequence variation, not just of the Sierran and lewisii and cardinalis, if we start looking at this whole group all across their genomes, it's clear that there's been a long history of integration and sort of mixing along the way, probably during different glaciations. There were times when they were separate, and then they came back into contact and exchanged genes at some loci, exchange variation at some loci, and then were separate again. And that's been going on for a very long time, including right up to the present. So if we go, it's not showing you this tree that I'm showing you, but if we go and look at Mimulus parishii, which overlaps with Mimulus cardinalis in Southern California, they grow right together, and we see hybrids in the field. Mimulus parishii clearly has a mitochondrion and a chloroplast that it recently acquired from cardinalis. Same thing if we go into the Sierras, we see that the Sierra and lewisii have a mitochondria and a chloroplast that they recently acquired from local Mimulus cardinalis, whereas the rest of the genome might primarily show a different pattern of variation. And so what we can do is not just phylogenetics, but phylogenomics, and ask how each chunk of the genome is sort of moving through space and time to sort into the variation that we see now. But that means that there isn't one true species tree, because the loci that control flower color, like these Mimulus cardinalis and the rupestris and verbenaceous down at the bottom, those might actually be totally shared, even though they're not as closely related genome wide as Mimulus lewisii and Mimulus cardinalis are. So not every gene in the genome is going to tell the same story. And again, I don't want to belabor it too much, but it turns out to be a much more complex, but also very interesting story. What we've also found is that there are chromosomal rearrangements, so changes in the whole structure of chromosomes that produce very strong hybrid barriers. So they create a very strong sterility barrier where hybrids are quite pollen infertile. And that is also contributing to them not merging back into being a single species where they're in range contact. We're also doing work looking at pre-mating barriers and the process, and how the barriers, the hybrid sterility barriers, may favor the evolution of even increased divergence in the flora morphology. And again, I'm not going to talk about this too much more, but this is ongoing work that we've been doing for the past 10 years, 15 years or so, and continue to do in my lab. So, but mostly we work on the next groups I'll tell you about. Okay, so working through the groups, so that was we're in the sort of water loving ones. And again, if you go up to Glacier, you'll see the Mimulus lewisii, that dark pink one, in every creek. Anywhere you go hiking at high elevation around here, you'll see that. And there's been really nice work actually done recently by Mackenzie Urquhart-Cronish, who's a grad student, was until recently a grad student at University of British Columbia, with Amy Engert, who's a Mimulus researcher, looking at patterns of genetic diversification as glaciers retreat and Mimulus lewisii moves into those new bare habitats. She actually came down here and did all of her genome sequencing for that. So she's looking at sort of bottlenecks that are associated with glacial retreat. And there's some nice papers coming out on that now. Okay, so that's the pink and red Erythranthe group. This next group is sort of a tricky one. These are the Paradanthus group. They have quite a lot of diversity, particularly centered around the sort of Snake River region, so and out to here as well. But these are the like sort of the least studied of the Mimulus in Western North America, and it's still really ambiguous what's going on with them sometimes. So some of them are perennials. So the picture, the sort of fuzzy one is Mimulus machadus. This is a picture I took down in Rattlesnake Creek. It's sort of just growing in the cobbles there. You can see it has a yellow monkey flower like flower, like the Mimulus guttatus in the background, but it's more radial symmetric, and it's really covered with these dense slimy hairs, which talk about the functions of those if you like. So those are perennials, at least probably not here. They're going to get washed out, but in many of their habitats. But then there are also a whole bunch of small annual ones in this group that are things like Mimulus washingtonensis, maybe ampliatus, and again the name and breweri. There's a bunch of these very, very tiny annual ones. And I just, if you were out doing Mimulus ID, I'm just sharing this. This is a nice, really lovely specimen. It's arc of flowers from the herbarium. I think it's a Klaus Lakshevitz specimen, so a very nice one. And it actually contains both what Mimulus washingtonensis, which are the ones that have the flowers open here. They have these kind of spade-shaped leaves. Now those are the three in the middle, but then these two that are flanking, those are actually Mimulus nasutus, which is in the next group I'll talk about, and a completely different section of the genus. But it's very hard to tell because these plants are like this big, and the nasutus are selfing, and so they don't actually make flowers that anybody would notice. And so if you're, actually the best way to tell these two sections apart, so the Parodianthus group and the next one, which I'll talk about in a minute, which are the other yellow monkey flowers, is sort of the calyx shape. The Parodianthus group ones like washingtonensis have sort of relatively parallel linear calyces where everything ends at sort of one point, whereas the other ones will have this sort of more I don't know, the top ridge of the calyx comes over, and they tend to be sort of more rounded. And that's actually and not all the calyx lobes don't end at the same point. And that's actually one of the best ways to tell if you have a random yellow monkey flower, whether it belongs in the N equals 16 group or the N equals 14 group, which is the guttatus group I'll talk about next. So we have these here, but they're obviously really ecologically diverse with just the two I'm showing you here, one being a perennial that lives in the gravel bars, the other being an annual that's, going to be in a little seep and going to be gone. I think the one in the pot here, I think I got the seeds up Mill Creek. So, it's growing like on the rocky spots next to the creek. So very, very dry habitat. So this is we might start working on this group, but it's we're waiting to, it is not yet well resourced in terms of genomics, but really interesting variation. Like there's Mimulus Jungermanioides lives in like the cliffs of the Snake River Basin, is super cool trailing plant. Okay, so last but certainly not least, I'm gonna talk to you a little bit about the Mimulus guttatus relatives. So that used to be section Simiolus, and that's mostly what we would call the Mimulus guttatus complex, but it includes a couple other things like Mimulus glabratus, which has its N equals 15 haploid chromosomes, sort of found down the middle of the country into Central America. But then with this Mimulus guttatus complex, these yellow monkey flowers being super common all across Western North America. So the map here is showing the range of Mimulus guttatus, so glabratus, which is very similar looking, sort of starts up just east of there. But then within this, there have been many, many other species, depending on how you count them, or populations that are distinct that have sort of blebbed off from within the sort of pool of diversity that is guttatus. And these, again, include things that live on really, really harsh copper mines, that live on serpentine soils, that live in Yellowstone, which I'll talk to you a little bit about, that live within the salt spray right on the coast. So there's tremendous daphic variation. So daphic meaning sort of adaptation to weird soils or harsh abiotic conditions. There's tremendous life history variation. So with, and by that we mean annuals versus perennials. So again, these are totally cost compatible and more closely related to each other than two annuals from different parts of the range. So this is an annual from Oregon and a perennial from Oregon, but so very, very different morphologically, but quite allied genetically, so tremendous life history variation, and again, repeated mating system evolution, where we have primarily outcrossing taxa, bee-pollinated taxa like Mimulus guttatus, evolving into these really tiny flowered selfers like Mimulus nasutus or laciniatus, which is in the Sierras, or Mimulus cooperphilus, which is on copper mines. So, and that happens repeatedly and independently over and over again. And so this group of plants has been a really, really, I mean, really important model for trying to understand sort of this kind of diversification. So how do you go from being a perennial to an annual? How do you go from being an outcrosser to selfer? How do you adapt to living on copper or serpentine or in the geysers of Yellowstone? And so that is what our lab and many other labs has been spending a lot of time on. Okay, so to remind you of those questions, we're going to be thinking about how my populations diverge. And I'm going to be focusing in a moment primarily on the morphological variation, which is probably what you're most interested in. Well, but I just want to emphasize that it's also really important to remember there's a lot of hidden variation, and that we don't always see all the variation that there is, even. So this is just a case of that where this is a single population we work on in the Oregon Cascades. All the yellow here are Mimulus guttatus. You see, it's a really beautiful wildflower site, but it's a hillside, you know, whatever, 30 meters across, 30 meters square max. These guys are mostly outcrossing, mostly pollinated by bumblebees. And this population is on record as pretty much the most diverse population of any species on Earth. So and that what that means is if you go into this population and you sequence individuals or make inbred lines and sequence those inbred lines, and then you compare them to each other and you look at their DNA sequence, two random plants from this population are basically have comparable sequence diversity, or we look at the whole population in terms of its diversity, to the sequence variation, so at the DNA level, that we would see across all great apes. So like humans, orangutans, chimpanzees, all of that. And so we're very tuned into the human primate variation, but it's really important to remember that you know, there's a tremendous amount of variation in some of these populations. And it is really an interesting question, an open question in evolutionary biology, why there would be so much variation, including variation at the sequence level, but also variation in things like pollen viability that we might not expect to be variable. And I'm not going to recap all the work that we've done to try to figure that out. But one of the things is it's just a large population. So again, it's one patch. There's another population a mile down the trail that's similarly diverse. But what work from John Kelly and others at the University of Kansas, and also work that we've been doing, really shows us that this is a population that lives off snowmelt. It's an annual population. And so basically the growing season, as long as there's snow melting and oozing down the hillside. And because of that, some years it's good to go slow and wait and make a bigger flower and more seeds. And other years it's good to go as fast as you possibly can because you're going to die if you don't set seed right away. And that varies from year to year, and the plants can't predict it because it's too late by the time they figure it out. And so what that means is there's this constant fluctuating selection where some years it's good to be a little bit big, some years it's good to be a little bit small, and that goes back and forth and back and forth and back and forth. John's done work over the past 20 years where you can actually see the alleles fluctuating that underlie very small differences in flower size, but that maintains this very large pool of variation. We've done very similar work showing that defense trait of glandular trichomes does a similar thing. About 80% of these plants don't make any trichomes, about 20 of them do. We think that what's going on is that in these longer snow years, the growing season is long enough that the plants actually experience herbivores, and then trichomes are valuable. But in most years, in dry years, they don't ever need those trichomes. And so if they make them, they're a waste. And so that again produces fluctuating variation, that fluctuating selection that maintains variation. The last thing that, again, I'm not going to dig into too much or at all, is that we also know that this very large population has a lot of hidden processes, hidden variation that are not obvious, like, or as obvious as things like flower size or defense. And so a lot of those are selfish genetic elements, and I'm not going to dig into this at all, but we've been working this population since the 90s, and it actually turns out to be a really, really interesting model for studying a whole set of processes that are natural selection that occur below the level of the individual. So not this plant makes more seeds versus that plant, this genotype makes more seeds than that plant, but this chromosome outcompetes that chromosome through meiosis, or this mitochondrion outcompetes other mitochondria in the population, and that is generating a lot of variation in the population as well. And that's a cool hidden aspect of this that I'm not going to talk about further, but I'm happy to talk about later. Okay, so from an adaptation perspective, I wanted to just, again, we are interested in many, many different aspects. So I'm showing you here some of the trichome variation. So some plants in that Iron Mountain population look like this, so covered in glandular hairs, and other ones are completely hairless. And actually, so that is under the control of a single gene. So, and so we're interested, and then that gene changes in frequency as you go down in elevation from that cascade population. So we're interested in things like that. We're interested in non-structural carbon storage and water stress and how physiological traits contribute to these things. And we're interested in all the aspects of reproduction, so life history, mating system, and phenology. And today I'm going to tell you two very small vignettes just because they're cool active things that we're doing that folks might be interested in. One having to do with the gain and loss of stigma closure, so a very macro scale question. So this actually goes beyond Mimulus, but we're using Mimulus as a way of understanding it. And then one from our work that's been going on for the past for several years, looking at how yellow monkey flowers have adapted to really extreme thermal environments in Yellowstone. So, and I have no sense of what time it is. So how are we in time?
it is 8:11.
Oh, wow. Okay.
But I
Okay.
think
Okay. So I will like show one slide from each of these. Sorry. Yeah, there is a tiny clock in there, but I'm used to having my phone and I didn't grab it. Okay. So I will tell you a tiny bit about stigma closure just because it's so awesome. And there, we might be able to watch a video. And then maybe I'll stop there and people can ask me about the Yellowstone stuff because I have lots of slides, but we don't need to talk about it. Okay, so stigma closure. And so this, again, I've been sort of narrowing down on this population level variation, but stigma closure is actually something where we sort of started narrow and then it turns out to be much broader. Presumably you all know what a stigma is, but I'll just say like this is a bilobe stigma of the Mimulus cardinalis I showed you earlier. It's a really nice close-up photo taken by Tim Wheeler. These are little papillae. This is where the pollen, which you can see being produced down below by these very weird spiky anthers, would ordinarily come from another plant on the bill of a hummingbird, and it would come and it would adhere to this, these in between these stigmatic papillae, and then the pollen, which are tiny microscopic, make that very long macroscopic journey down to fertilize the ovules. So the stigma is really this place of interface between the outside world and the very personal internal world of the plants making its own seeds. Okay, so it really is the pollen has to come from the outside and go to the inside. So it's really a very important but very, very understudied and poorly understood component of sort of flowering plant reproduction. So if you care about seeds and crops, you really want to understand what happens at this interface, because this is a gateway between the sort of pollen and the pollinators and the actual seed production. So, and in Mimulus, it has this other cool feature, which is that it involves touch sensitivity and rapid movement. So, so here again is the cutaway of the flower. Again, in Mimulus, we have this bilobe stigma. So like this. And hopefully I can show you. So the one, it's an irritant if you're trying to do a lot of crosses, which I am. So in the outcrossers, this is touch sensitive. So, is it going? Yeah. Okay. Whoop. One last whack there. Okay. This other one, this is so that was touch sensitivity. You can replicate that by doing, this is under, you can pull a vacuum. So it's a controlled wilt. And this is just like our own nerves. Okay. So this is an electrophysiological trait like Venus flytraps, which catch insects, and sense the flies walking on them, and that's how they close. Or like the nerves that we have in our bodies. Okay, so this is sort of unusual. We think of plants as being pretty slow. So there's Mimosa pudica, it does a similar thing where it folds its leaflets when it's touched. And so, but there's a relatively small number of these kinds of rapid movements in plants. And so we're really interested in this, again, in its found not just in Mimulus, though it's found pretty much in all Mimulus. It's found all across the core Lamiales. So this picture, the other video here is a plant known as Proboscidea, or devil's claw. This is in the desert Southwest. It's quite distant from Mimulus. It's in the Martiniaceae. It's got a fruit like this big, looks like a big okra with a hook on it. And the flower is kind of Mimulusy, but you can see it's got a similar looking stigma. And again, if you touch it, it closes up quite quickly. And so this is really fascinating. It's been something that's fascinated botanists for hundreds of years, really. Not sure what those ads are about, but let's go to the next one. And so it's really, really interesting. And it's also been sort of almost totally unstudied. So we have new funding now to really try to understand what is going on here, both because it's super cool and interesting and to think about how that rapid movement is mediated, but also because of the second thing the plants do, which is if they're touched, they close, but then they reopen, the stigmas reopen if they haven't been successfully pollinated. So if they didn't get enough pollen, if they got sterile pollen, if they got pollen from a species that's too distant related. Okay, so if they get bad pollination, or I just touched them and didn't provide any pollen at all, they reopen. So it takes about more like 20 minutes to half an hour, and they've reopened fully. They also become less sensitive over the flower age. So like day one flowers, the day they open, are very touchy and close quickly, and older flowers close more slowly. So there's clearly some fairly subtle modulation here. But what the reopening tells us is that not only is there's this sort of rapid touch sensing, there's a slower, and I think in some ways even more fascinating, pollen sensing. And the plant has a way to count, so the quantity, and assess the quality of the pollen that is on that style, because it only reopens if it didn't get a good load. Now you might be like, oh, well, that's because its seeds are being fertilized. The pollen has barely germinated within that 20 to 30 minute window. It takes hours for the pollen tubes to grow all the way down and actually fertilize the ovules. So the plant is sensing at a very sort of at the stigma scale whether it's been successfully pollinated. And that's just really fascinating because it provides this window into this sort of hidden communication between the male and female components of plant reproduction that we really we don't know enough about, I should say. So somebody may be wondering why they do this. Why would a plant do something fast? So plants are not stupid. They could be fast if they wanted to be. Mostly they don't need to be. They're eating light and water, and they're sort of not fast moving things. But here they're interacting with animal pollinators, and animal pollinators do move fast. So that closing is happening within a pollinator visit. And the two reasons they might be doing it have to do with getting out of the way so they don't accidentally self-pollinate, so that they only receive pollen in, but also preventing pollen being knocked off as the insect or bird leaves, to increase male pollen export, as well as successful outcross fertilization. So there's been some work done on that I won't go into. And then again, this reopening is really fascinating, because the fast part's cool, but the reopening says that the plant knows somehow how much pollen it's gotten and whether it's the right pollen that it wants to sort of refuse to be open again for any more pollination. It's done. And I should say nobody really knows yet, but we're trying to figure out how do they sense both the touch, the sort of fast electrophysiological thing, and then the slower pollen sensing process. And we're able to do that because, as it turns out, going back to the mating system things, there's been repeated evolutions of self-fertilization within Mimulus, and the touch sensitivity has been lost repeatedly. So independently in Mimulus nasutus, which is the selfer, Mimulus parishii, which is in a totally other section, which is also a selfer, also in the N=16 group. So these are Washingtonensis and Patulus, which is a selfer, some really nice but unpublished work by Robert Menke demonstrating that. And so it looks like it's very tightly associated with, once you become a selfer, it's probably not a great idea to close because you're trying to self-pollinate. And so they lose the ability to do it. But that gives us a genetic sort of what is the word? Like screwdriver to sort of get in there and try to figure out how it's working. And so we have a lot of, I'm not going to belabor this, we have a lot of ongoing work on this. So using lots of comparative genomics across all Lamiales, actually, looking at what genes are being expressed in stigmas that can and cannot close, doing lots of traditional genetics, and then also spending some time in the field trying to figure out how do pollinators interact with these different strategies. And that's, again, a super, super awesome trait, which we're taking lots of videos of. So that's the other thing is Yellowstone stuff. But in the interest of you all not having to stay up too late, I'm going to stop. And we are basically, I'll just say that yellow monkeyflowers, Mimulus guttatus, get into Yellowstone National Park, where within the park's boundaries, they basically completely recapitulate the range-wide variation I'm showing you here, where they have adapted to this sort of complex mosaic of soils generated by the geothermal effects. And that involves both below ground adaptation, so the kinds of things we saw with copper tolerance, life history variation. So in thermal areas, they evolve to become annuals. They might be perennials, a meter away in a different habitat, and that's genetic. And they have evolved increased selfing rates and shifts in the photoperiod control of flowering, because in order to live in the thermal areas, show you some pictures of that. In order to live in the really extreme thermal areas, they're basically shifting their entire life history to be in the wintertime. So they're like out there photosynthesizing now, the snow falls, it hits the hot soil, it melts, the plants are exposed. As long as they stay very close to the ground surface, as you can see them doing right here, so they're genetically dwarf as well, they can reproduce as long as they can self, and as long as they can flower under the short days of spring. And so they've done all of those things on a scale of meters that we see happening across elevational gradients elsewhere in the Mimulus guttatus complex, which is super awesome. And we've figured out how they've done it. And I can tell you more about that if you're interested. So in the interest of time, though, I'm just going to, so many slides. Yeah, I should say thank you, and thank you all the people who've contributed to this research. Thanks to Alexandra for inviting me and for introducing, and I apologize for running so far into the time here. Should have kept better track.
Lila, we're gonna need a sequel.
Yeah, sorry, I should have probably
Lila,
just talked about the Yellowstone thing. I mean, I give a whole 50-minute talk on Yellowstone, but I kind of want to talk about the rest of it too. So yeah, could do this. So please, if you have any questions and are not asleep or home somewhere else now, ask.
We have some questions from throughout that I'll go back to, but please, everyone, enter into the chat, and we can, as long as Lila's willing to answer.
Oh yeah, yeah.
So the first question was from Karen Porter. Does the plant sense the absence of pollinators and quote unquote decide to self-pollinate? Is the ability immediate or developed over time and irreversible?
So yeah, so in the selfers, or let me see, go back to some pictures of flowers so that it would help. No. So the differences I'm talking about here are genetic differences between species or populations that are manifest in differences in floromorphology. So if we go back, say, if we look, you guys see this here. So Mimulus guttatus is large flowered. It's attracted to bees, mostly pollinated. It has nectar, scents, mostly pollinated by bumblebees out in Oregon, also here. And then these self-pollinating taxa, the Mimulus nasutus I'm showing here, which looks like it just has buds, those are full-size flowers. So especially if they're grown under any kind of dry conditions, so there is a somewhat plastic component, they don't ever even open their flowers. So the male and female parts are touching inside the flower, and the pollen is released before it would ever open. So when I want to do crosses with them, I grow them in a cool growth chamber, and I give them short days, and I make them think it's wintertime, and they have forever, and they don't need to hurry up and self. And then I can get them to make a flower that looks more like this second one here, which I can handle and do a cross with and emasculate to do crosses. But the differences I'm talking about here are, fairly extensive evolutionary differences. But again, it is also the case. So I don't know if you like impatiens. It's more of an East Coast plant, but violets do this. Some plants make chasmogamous flowers, which are open and regular normally pollinated flowers, and then also what are called cleistogmus flowers, which are like these Nasutus buds that never really open, and they'll make them on the same plant. And those they do tend to they'll like impatiens that's in the shade might make more of the selfing ones. And so there is a plastic component, but these are, in the case of the ones I'm talking about, they're evolved strategies. In the Yellowstone one, it's an evolved strategy. The really key trait is reducing the stigma anther separation, which is the distance between the pollen and the stigma. And so if that distance is, say, negative, where the pollen is being released above the stigmatic surface, it can get on there. But if it's well below, like it is in these other ones, or in that red one I showed you, I can show you. Yeah, so this red Mimulus cardinalis, the pollen is below the stigmatic surface. It's also, in this case, usually released a little before the stigmas mature. And so, and these are long flowers that are attracted to hummingbirds. And so the hummingbird comes in with pollen from another flower on its forehead or beak, and it hits that style stigmatic surface first, that closes, and then it picks up pollen for its next trip. So there's always an in and an out in those outcrossers. And so so these are genetic things that affect them, that are manifest through the morphology. But there's also a plastic component. So I've been working these Mimulus cardinalis I have in the greenhouse, and I feel like sometimes that stigmanther difference is quite large, like half an inch, and sometimes it's relatively small in the same exact genotype, maybe depending on like whether it's hot or cold. And if it's hot, it might want to self more because it knows it's going to be summer and it's going to die. That's totally hearsay. That's not, I don't have rock solid data. But I suspect that there's both, but certainly plenty of genetics. Does that answer the question?
I think so. And there's kind of a bit of a follow-up question from Lisa, um, asking if the diplicus color polymorphism is driven by pollinators, kind of a similar, yeah.
Yeah, that is almost certainly not driven by pollinators. And I should say, like, again, there's pollinators also see differently than we see. So there may be UV polymorphisms in a lot of these taxa. There are in some of these. So, but that pink yellow, those are both bee. Those are both bee pollinated, and pollinators have been shown to move freely between them. What is probably going on there, the pink has to do with anthocyanin production. So this is like your red cabbage also is or your blueberries. This is why they're antioxidants. And so this is why you're supposed to always be, eating your red fruits. And so that pink yellow may be under some kind of fluctuating selection where the pink is favored on worse soils or in droughtier conditions, and the yellow on others, but it's enough of a variable situation. And there's a seed bank that you see them in these mixed pollination populations. And I say that because there's really classic work going back to like the 1930s on what used to be called Linanthus perrii. It has a blue light flower color polymorphism in these similar Southern California deserts. And there's really nice work in that case showing that the white one is favored in wet years, which occur every 10 years, and the blue one is favored in worse soils in drier years. But there's enough variation and enough mixing that it's just a balanced polymorphism. So in that case, it's almost certainly not about pollinators, but about a sort of a side effect of wanting to have more of these anthocyanin compounds to deal with some kind of abiotic stress. But people haven't done that study, but that's what I would think based on the other work.
Laura was wondering, are you aware of examples where humans have brought Mimulus species, ornamentals or crops into an area, and these new species influence the fitness of the local populations?
Okay, yeah, so monkeyflowers are invasive in New Zealand and Europe, and there is really interesting dynamic, and also on the East Coast to some extent, where they're not. So that's particularly Mimulus guttatus group. So I didn't have time to talk about. So the Mimulus guttatus group is mostly diploid and 28 chromosomes here, so 14 pairs. There's an offshoot of them in South America that's tetraploid, and they also have pink yellow polymorphisms, which we don't see anywhere else in the guttatus group. And then those were moved to, so both of them were introduced into the British Isles and formed like hexaploids. And so like if you go down to your Keris nursery or whatever and buy, Mimulus, they're often these sort of spotty multicolored ones. And those are actually complex hybrids of like the South American ones with other ones in Europe. And so so in that case, I don't know if they're um so in that case there aren't any native Mimulus in Europe, but there's certainly interesting interactions when you bring multiple genetic entities into contact in a new place, and they can hybridize. And in that case, I think they just clonally reproduce in streams all over Scotland. There's Mario Vallejo Moran, who's I think now in Sweden, but he was at Stirling University in Scotland. He picked up the Mimulus bug when he was a grad student at Duke, and I was there, and so he studies all these weird polyploids in Europe. But yeah, so it's hard for me to think of them as being invasive because they're so lovely, but they're all over Europe, and it's sort of complex hybrids. Yeah.
Fascinating. And Larry Holdsworth was wondering, how do you account for the miniature monkey flowers with a plethora of colors in the craters of the moon?
Oh, how do I account for them? More power to them. So yeah, so yeah, we haven't collected there ourselves, but my grad student went down there actually last year, and so so a lot of those are the sort of what it's been split up a little bit, are these sort of Nanus ones that are and those have relatively big flowers, but there's also I think Sextorphii and Breweri and a couple of these other really small sort of annual Paradanthus type ones, the sort of N=16 group. I would say Mimulus are really, really good the one thing that they don't do that well with is competing with other plants. So like you don't see them growing in forest understories. And so like and that's like true for Yellowstone as well. It's a whole lot of open habitat. Almost nothing lives in those thermal areas. And so they're really good at adapting, not necessarily to tolerate the super high stress conditions, but to kind of avoid them by flowering super early in the spring. So they're really adaptable and at getting into their niche, which is like ephemeral water in the spring, by being annual in those places where they have to be annual and flowering early where they have to flower early, not necessarily by tolerating and living through the whole horrible, burnt summer. So I would say there's lots of them there because like they can be there, and they're not going to grow in a grassland really well,
Mm.
generally speaking. So they're not, you don't see them in sort of a dry grassland. But if it's a dry place that has nothing else growing in it, and they can come up early in the spring and make a ton of flowers, then yeah, they have the sort of genetic variation to adapt and do that. But
Mm.
people haven't studied the Nanus are really interesting. There's this sort of carpet of purple down there in the spring. It's amazing with maybe a few yellows here and there. But Nanus, it sort of in the old sense is like has this huge range and like I was collecting some out in Oregon for a collaborator recently, and there's like 3 or 4 different species named like within Bend, like a park in Bend. And so I don't have a sense of like how much of that variation is different species versus just free gene flow between different genotypes.
Mm.
So I don't know if that counts for it, but I'm not surprised that they can do it.
um, so Beth Madden wants you to go back to something you mentioned. What's the story with the dense slimy hairs on the
Uh,
Mimulus muscata in Rattlesnake Creek?
Well, I mean, so Machatus, I mean, or
Machata.
whatever you want to call it. But yeah, Erythrinthe muscata or Mimulus machatus. That means like slimy. So like the moss roses, which are also are Rosa machata, it just means slimy. So I don't know that anybody studied it in those ones in particular. We study very similar traits that are variable within the Cotatus group. So actually, like this little rosette here is covered in and this plant would be slimy if I touched it. Um, so I think it's different in different settings. So and Mimulus tillinghii, which is the yellow Guttatus-like one that grows up at high elevation. So you see it with Lewisii up in Glacier. So they often are sort of co-occurring up there. They're often slimy as well. So in some of those cases, like the high elevation ones and the ones in Yellowstone, we think that it has to do with abiotic stressors. So both sort of dealing with cold, but also dealing with high light in those habitats. But in Oregon, where we're studying similar trait, slightly different genes, but same glandular trichomes, we think it has to do with variation in herbivory. So I think in the species like Machatus, where everybody's super slimy, it's probably a long-term adaptation for some kind of abiotic stress. But and may not hurt from a biotic perspective as well. But in some of these cases where there's polymorphisms, that probably has to do with more with variation in herbivory pressures from year to year. So different in different cases probably. In Yellowstone, we demonstrated that they have the most extreme annual populations are covered, like the young leaves, but not the later leaves, are totally covered in slimy hairs, and that can't be herbivory because they're making those leaves in like February, like an inch from the ground in the middle of Yellowstone when there's meters of snow around, and there are no herbivores, like no insect
Mm.
herbivores are flying around and landing on them. But we showed that they shed snow, so the sort of slimy hairs cause water to beat up and run off, and that probably helps them do their photosynthesizing, despite the fact that they are constantly being snowed on in near the Continental Divide. So again, it's a little bit
Wow.
different. So I don't know for sure what's going on with Machadas, but we have evidence of both abiotic and biotic adaptation.
Wow. So one adaptation can be useful for many different things.
Yeah. Yeah. And I mean, where I'm working on a paper now where it's also, they're very modular. So, I mean, and the reason we're a little bit skeptical about some of the taxonomy is some of it is based on things like hair morphology. But every time we try to understand the genetics, it's like a single gene turns those hairs on or off. And that's not a good way to define a species. I mean, it might be characteristic of a species, but it's not like a species barrier, that one gene. There's things that are very, very easy to turn on and off. And so, yeah. So, but what we've also found is that it's very modular. So there are different genes that control like leaf hair, like hairs on the early leaves versus the late leaves versus the calyx, and glandular hairs versus non-glandular hairs. And so it's really the plants aren't like, oh, I have to be hairy all the time. They're like, oh, I want to have early hairs, but not late hairs because of these conditions. And so they're really good at sorting that variation out depending on what the natural selection is locally.
Wow.
Um,
Um, so incredible. I think that's all of the questions that have been that brings us to now, but
okay.
we still have quite a few people in the room with us. Does anyone like to raise your hand? There's a raise hand function if you want to ask a question, or you can ask into the chat. And I don't know, Lila, if you've been reading the chat, but there's a lot of really
Oh yeah, people are so sweet.
complimentary
That's so nice.
complimentary
I'm so
things.
sorry for running over. I knew I should have.
No, no, I just want you to note that so many people really enjoyed this. One person said in particular, um, let's see, can I find that? Um, Oh yeah, here Alexia Cochran said, "Thank you so much, Leela. People like you and our own are why Missoula rocks."
Okay. Well, I'm just doing my science here, but it is. I mean, it is. I mean, it's hard not to like love the plants because they are. I mean, they're so. I mean, yeah. It's just really interesting. I mean, I've always been a plant person, but I teach the flora class here mostly to wildlife biology students, and I do really try to convey because again, a lot of people they just see green and they're like, green stuff, plants,
Mhm.
elk eat it maybe. And so, I mean, that's not fair, but it's really, I mean, there's a tremendous amount of diversity. That's this sort of very fine scale tapestry that's really nice for people to appreciate. So, um,
Yes. Yes, absolutely. Well, I do we might have to have a part
yeah,
2
sorry,
next
you probably billed
season.
it as me talking about Yellowstone stuff, but
No, no, please don't apologize. This was amazing. And I'll just give people one more opportunity to enter a question. But yeah, I do we're really grateful for your time and your expertise. It's just I mean, just all of these photos are so incredible to look at.
oh yeah, well, I mean, yeah,
You
that's
promised pretty
thank you.
photos and you
I
delivered.
did. And some of them I took myself. I don't get in the field as much as I'd like, but I get in the field some. And
Yeah.
you have them in the greenhouse too. So yeah, if there's are there any other kinds of questions?
It looks like we've just got a lot of people thanking you.
Okay, well,
So,
thank you. Thank you for being patient. Um,
yeah.
and you could Google Fishman Lab, and if you and email me if you have a question that comes up later, because I'm always happy to talk about the plants.
Oh, well, thank you so much.
Okay,
And
thank you. And